Thank you all for coming today. So I'm going to start by talking a bit about functional connectivity inference. And more generally speaking, in fMRI, the topic of our inferential methods have come under increased scrutiny in the recent years. This is actually a really great thing for the field because more accurate inferential methods will lead to more reproducible and more robust findings. So I'll talk a little bit more about functional connectivity in particular. So the most focal level of inference that we can use is the individual edge level, or the level of individual connections. However, this can be very conservative in the sense of leading to many false negatives. In part, that's because there are so many edges, around 10 to 100,000 edges. So we have to correct for that many comparisons, and there tend to be very modest effect sizes. So this has been shown by Merrick and Turbo-Clemens and colleagues recently. This problem has been partly addressed in the field of fMRI through a procedure called cluster level inference, which groups together information in the local neighborhood. So in the activation mapping context, these are local clusters, as you can see on the left of this figure. And in the functional connectivity context, these are edges that are connected by nodes. So this has been translated from activation mapping to the network context by Zaleski and colleagues using the network-based statistic. However, these approaches can lead to a sort of tip of the iceberg style effect and low power at standard sample sizes. So the figure I'm showing here, we and others have found that using the whole Human Connectome Project dataset, we tend to see these very widespread diffuse activations. And the Creamer's study shows what happens when you subsample smaller samples, you tend to get these more focal activations. And we see something of a suggestion of this phenomenon in the context of functional connectivity, where Siwan Gao and colleagues and Faskowitz and colleagues have found that when you group together edges, you tend to see these widespread groupings that seem to be reproducibly obtained. So could we move beyond edge and cluster level inference to broader levels of inference, and would this improve power? So I'll introduce a couple broader levels of inference. The first is called a constrained network-based statistic. More details are in this manuscript, but in brief, you're pooling together information within predefined subnetworks. We have a predefined partition. Then we non-parametrically estimate the null for each of these subnetworks, and then we do a parametric multiple testing correction. So this has a couple neat features. One is that there's no contiguity requirement. So we don't require edges to be touching nearby nodes in your data that are above a certain threshold. Instead, we're pooling across these predefined areas. And there's no assumption that statistics are identically distributed. We're actually estimating null uniquely for each network. We've also created a multivariate extension of this called the multivariate CNBS. This combines information across the whole brain. And also this only uses a single test, so there's no need to account for this multiple testing correction. So I've talked about different levels of inference. How can we benchmark the power of these? So here we're using 8 tasks from the Human Connectome Project, 7 kind of real task contrasts, which are a task versus rest contrast, and 1 kind of pseudo task contrast, which is rest versus rest, where we shuffle rest 1 and rest 2 identities for each subject. So we start with the full sample size for each of these tasks. And then at each of 500 repetitions, we randomly sample 3 group sizes. And then we perform some statistical inference. I've listed the 7 procedures we'll be using here at the right. 2 edge-based procedures, 2 cluster, 2 network, and 1 whole brain. And then we can acquire the significant results at each of those 500 repetitions and pool those together. And we can compare where we've seen significant results to the ground truth obtained from the whole sample size. So we can estimate Cohen's d at each of those edges. In areas that we see a positive effect, we can see whether we also saw a significant result in that direction. So we can combine the significant results and the ground truth to estimate true positive rates. And just as a reminder, we're doing on the order of 170 benchmarking experiments. That's 8 tasks by 3 group sizes by 7 statistical procedures. So we'll start by looking at the ground truth. So this is what the ground truth effect sizes look like at the edge level. And when I estimate which edges are significant, for each of these tasks, we tend to see about 87% of edges are significant. We can also take a look at the distribution of results at the cluster level. I'm applying different cluster determining thresholds based on the Cohen's d. Here's cluster determining threshold of a small Cohen's d effect size of 0.2, and we tend to get just two clusters, one positive cluster and one negative cluster. And if I increase the cluster determining threshold, I still get one positive, one negative, all the way up to a large Cohen's d of 0.8, one positive, one negative cluster. So just these two clusters that span across different areas of the connectome. And only until we get to a very large Cohen's d of 1 do we start to get clusters that seem to separate out. So suggesting that really at most Cohen's d thresholds, we're seeing these very wide distributed clusters and less so focal effects. We can also take a look at the network level. This is pooled Cohen's d, or rather Cohen's d from pooled connectomes at each of these networks. I can also look at which of these networks are significant after multiple comparison correction, and almost 100% of these networks are significant for the average task. So altogether, these results suggest that there's some widespread ground truth effects rather than more kind of local effects. We can also look a little bit more specifically at the network. We estimated the variability between these networks using kind of a non-parametric ANOVA approach where we shuffle node community membership, and there was significant variability. I just want to highlight here that shows some support for the meaningfulness of these networks. I also want to highlight that we find within communities, there tends to be a decrease in connectivity in task versus rest, suggesting perhaps some sort of increased integration across communities during task relative to rest. We also, just so you know, we looked specifically at the non-parametric ANOVA just within these groups alone, and just this diagonal also shows substantial variability between each of those within community pairs. Just letting you know, the individual tasks show a very similar pattern of results as what I'm showing here, which are the average task results. And we can also look at the distribution of effect sizes at each of these levels of inference, specifically edge, network, and whole brain. And here I'm highlighting a medium effect size, and edge level effects tend to be smaller than medium, whereas network level effects tend to be a little larger than medium, and whole brain level effect is larger than medium. Remembering we're combining both large positive and large negative effects for the whole brain. So if we switched out those two things or separated them out, we might see even larger effects. So just broader scales tend to give larger effect sizes. So now that we've discussed ground truth, we can look at power after resampling, after subsampling. So here's the average power to detect an average effect at a group size of 40. And this is the error bars are showing the standard errors of means of across tasks. And this is how results change when we increase the sample size. So you'll notice that we only achieved what's conventionally considered to be adequate power, 80% power, for more global approaches, for the network-based and whole brain approaches. And we can also break down these results by individual effects. So here I'm showing on the x-axis different power levels, and on the y-axis, that's the proportion of effects that achieve that power level or greater. So here I'd like to highlight that the edge and cluster level approaches, only 25% or fewer of the edges in the connectome achieve the desired 80% power level, which is that dashed yellow line here at 80. So just sort of highlighting that at really standard sample sizes of 40 subjects or below, we tend to miss 3/4 of the connectome when we use edge or cluster level inference. And when we increase sample sizes, of course, everything again improves, and the difference between approaches starts to become smaller. And a final thing I'd like to highlight here is that we see a strong advantage of FDR controlling procedures over the other procedures that are designed to control family-wise error rate, such that even the edge level procedure that controls FDR outperforms the cluster level procedures, which are both designed to control the family-wise error rate. All right, so we can take a look at how each of these levels of inference affect the effect size power relationship. So on the x-axis is effect size, and we see that power improves with effect size as we'd expect. But something I'd like to note here, the two curves that are all the way to the left are both FDR controlling procedures. And what that shows is that for a given effect size, we have much better power to detect these with the FDR controlling procedures compared with all the others. And again, as we increase sample size, everything gets better and the difference between procedures narrows. But just highlighting that for FDR, it's like having twice the subjects. If you look at the curves for 80 subjects compared to 40 subjects, for instance. We can also look at how different areas of the connectome might contribute to better power. So again, we know increasing effect size leads to increases in power. What else contributes to power beyond the effect size? So we can take these curves and we can actually look at the residuals of the fit to these curves. And that's what I'm plotting here. And we can look at these residuals on the connectome. And I'm just going to average within network pairs for everything. So we can see for the edge FDR controlling procedure and the cluster controlling procedures, we seem to have a slight bias towards detecting effects in subcortical and cerebellar areas compared to other areas. If, for instance, the effect size were the same across the whole connectome, then we'd see more effects there. But this happens a little bit more at smaller sample sizes than larger sample sizes. So here are the results at the 80 subject and 120 subjects. We tend to see less of this bias. All right, so we've talked a lot about power. Another really important thing to know about each of these approaches is whether they control familiarized error rate to expected levels. So here's the 95% confidence interval for familiarized error rate given the 500 repetitions and expected control at the 5% level. We see actually that all approaches achieve valid familiarized error rate control. That is, they're all below the upper bound of the confidence interval. However, all these approaches tend to be on the conservative side, except for the network level inference. And I'd like to specifically highlight the whole brain level inference, which actually previously we always detected true positives for the whole brain level inference. We never missed an effect, but here we're never detecting any false positives in any of the experiments we ran. And just a reminder, this is REST 1 versus REST 2, but shuffled for each subject. All right, so just to summarize, we've introduced some new methods for broader levels of inference that address limitations of existing cluster level inference methods. We show evidence of meaningful network level effects, which supports the utility of the network level inference. And we find better power generally with broader levels of inference, although we're sacrificing some spatial specificity when we get to these broader scale inferential approaches. So a quick discussion of some implications to consider. Implications for functional connectivity. We might think a little bit more about the meaningfulness of different scales of inference. I'm just going to show this figure here. We find that the network and whole brain level inference tends to capture this underlying dependence in the data, the underlying structure, a little bit better than edge and cluster level inference. However, we're trading off, especially with the whole brain level inference, our ability to localize effects. So I think the network level inference strikes a nice balance between localization and capturing some underlying structure. There's also points to the promise of other multivariate procedures, both statistics and machine learning. We found some interesting things about network integration during task with the ground truth effects. I'd also like to point out that we actually, I was inspired for this study by looking at the activation mapping context. And I think that this study also has implications back towards the activation mapping context where cluster-based inference is very commonly used. And I'd also just like to point out that we're leveraging larger than ever data sets to inform typical studies. And that's something that seems to be a trend in the field recently. So I'd like to thank my postdoc lab led by Dustin Shinost, the HCP project, my funding, and my collaborators.
Hello, everyone. Welcome to my OHBM 2021 talk about all-resolution inference. I am Jonathan Rosenblatt from Ben-Gurion University in Israel. I'll start by thanking my colleagues, Yele Goman from Leiden University, Wouter Wida from Leiden, Livio from Padua, and Aldo Solari from Milan. Now, our motivation was twofold. First, what we called the spatial specificity paradox, the observation that the larger the cluster detected, the less we know about the location of activation. That's simply by observing the hypothesis being tested using random field theory. The null hypothesis is that there's no activation anywhere in the cluster. So by logical implication, if a cluster is rejected, it means there is at least one voxel with effects. So the larger the cluster, the larger a rejected cluster, all we know is that there is at least one active voxel in the cluster. So the less we know about the localization of activation. The second motivation was the observation that there's always multiple dippings of the data. For instance, because you vary the cluster forming threshold, and that will create multiple dippings, or because you changed your inference criterion from height p-values to volume p-values, right? Or maybe you change the smoothing of your data set. So even if you try very hard, it's just the assumptions required for the usual random field theory inference or other types are just too stringent. So we would like something that is valid even when multiple dippings have occurred. So essentially, we'd like two things. We would like, for any given set, doesn't matter how it was defined, so maybe a contiguous cluster or not, could be two hemispheres, whatever. We would like to estimate the proportion of active voxels in that set. We call it the TDP, the true discovery proportion of the set. And we would like our bound of the TDP to have like valid statistical guarantees no matter how the sets were defined. So even if you peeked at the data and peeked at the data again and really tweaked and optimized the form and shape of your set of voxels, we would like the guarantees on the estimate of activation of proportion of activation. We want the guarantees of this estimate to be valid. Now, the solution, we call it all resolution inference, ARI. It essentially returns lower confidence bounds on the proportion of true activation. And again, these bounds are valid no matter how the clusters were formed. Now, an example, we used the Bertrand et al. Dataset from 2015. It's an auditory task, so the subjects were requested to distinguish between a vocal and non-vocal stimuli. And you see here a set of clusters, all significant, created using a cluster forming threshold of 3.2, and all significant using random field theory. And in the last column of the table, we apply our estimator, which seems to suggest that the clusters may be significant, but most of them are not active voxels. So most of the voxels in the set are not. And you may wonder, how can our estimator be 0 if the cluster is significant? Well, the assumptions underlying cluster inference and our own inference are not the same currently, so these two methods need not agree. So this is the type of result we're aiming at. Here's another demonstration using the Lee et al. Dataset. This is a go, no-go task, so subjects were asked to press a button if the face that were shown was a happy face. And you see here two clusters, A and B, both formed using a 3.2 cluster forming threshold. So these are fairly large clusters, and we apply our estimator, again, in the rightmost column of the table. And you see that most of the cluster does not seem to be active. A third maybe in the first, in cluster A, and a half in cluster B. So we would like to find those voxels in the cluster that are driving the activation, to localize them, to drill down, right? So what we could do is raise the cluster forming threshold, say from 3.2 to 4. This will naturally return smaller clusters by definition. Some clusters may even break to subclusters, which is indeed the case, as we can see here. Now, what we gained is that when we estimate the true discovery proportion in these subclusters, they tend to rise, right? Because we're focusing on mostly active sets of voxels, and indeed, you can see that cluster B was broken down to various subclusters, and in this large yellow blob, it consists of almost 1,000 voxels, and 85 of them, at least 85% of them, are active. So this allowed us to localize the activation within the large cluster B to the yellow subclusters. And the same happens in cluster A, where we are able to localize the activation to the orange subcluster, number 1. Now, what's the catch, right? Because I'm showing you an estimator of a reasonably interesting quantity, which is valid no matter what you do, right? This is pretty much goes against everything we're taught. So what's the catch? Could it be in the assumptions? Now, I say that no, there's no catch there. What you need, we have the proofs, and what you need for the analytical proofs is what's known as the PRDS assumption, the positive regression dependency on a subset. And everyone who does FDR control, false discovery rate control, using the Benjamini-Hochberg algorithm, implicitly believes in this assumption. So this assumption is everywhere in neuroimaging, and it's essentially stating that the field is not is smooth in one way or another. It's actually less stringent than the random field theory assumption, because we're not assuming Gaussianity, and we're not assuming that the spatial covariance is stationary or isotropic. It's a broader class of distributions. Now we have the analytical proofs, but we also verified it using various NeuroVol data sets that's soon to be published. And indeed, we can state confidently that we do not have type 1 error rate inflations. Again, type 1 error when discussing confidence intervals means that we're not overstating the true proportion of activation. Power may be a concern, right? Because I could be overly conservative and say that the estimate of, for each set, estimate the proportion of activation is 0. And this would be correct because, it will always cover the true proportion of activation as promised. Now, to verify that we did not lose too much power, we did the following. We revisited the Pernay 2015 dataset, and we used the usual 3.2 cluster forming threshold. Now, we applied our TDP estimators. So we estimated the proportion of truly active voxels using a subset of 33 subjects. And then we validated it on the holdout of 66 subjects. Now, what is validated means that we revisited the same clusters and we counted what is the proportion of supra-threshold voxels in the holdout dataset. And here's the result. You have here various regions at 3.2 or 4 cluster forming threshold. In the third or fourth column, you have our estimator of proportion of activation. And in the last column, rightmost, you have the validated proportion of activation. So you may see that for the larger clusters, our bounds are fairly tight. We estimate the true proportion of activation to be around 78%, and our validation is roughly 84%. Same goes for this 97% of activation, which validates into 98% of activation. So the larger clusters are okay. The smaller clusters, our estimates seem to be overly conservative. We estimate that in these clusters, right, there's no activation, and when validating, we do find some activation. Now, I need to warn against the intuition that that the larger clusters return tighter bounds. We're still looking into the matter, and it's not an intuition I can stand behind yet. But again, where it matters, certainly in this case, the larger clusters where we have more spatial incertitude, at least there we have tighter bounds. So we can say better what is the true proportion of activation within the cluster. Now, for the practitioner, I guess you should be wondering, how can you use this and why does it work? So currently there is an R implementation in the RE brain package. We use the RNIFTY package for handling brain volumes and the HOML package for an efficient implementation of the estimator. Python users, the NiStats import has the cluster level inference, which already implements RE. And hopefully FSL, SPM, AFNI, and Brain Voyager users, we're working on contacting the developers and implementing for you, but this is still work in progress. Now, how does this work? For the technical details, I'll reference the 2019 Biometrica paper by Goman et al., simultaneous control of all false discovery proportion and large-scale multiple hypothesis testing. But in a nutshell, I will say that, first of all, you need some sort of assumption on the structure of the noise, right? Because you want to be able to distinguish between signal and noise. So you need to assume something about the well-behavedness of the noise. In our case, it's the PRDS assumption. You could replace it with some other assumption, a smoothness, whatever, and the algorithm would need to adapt accordingly. And the estimator, it's a confidence interval, right? So you may know that you can construct a confidence interval by inverting a statistical test. And that's what happens here. We took a class of tests called closed tests, which are very powerful tests that give you family-wise error rate control. The difficulty is usually a computational one, but they're guaranteed to have good power. And we inverted this test, and that's why the confidence bounds are fairly tight, as our validation has shown. And these are essentially the two secret ingredients of the method. In conclusion, all-resolution inference allows us to estimate the proportion of truly active voxels in any set, no matter how the set was constructed and selected, even after infinite circularity. This flexibility comes with a price, power. I hope I convinced you that the power loss is not terrible. And also, there's a lot to be said about changing and adapting and improving the method so that even less power will be lost. There are currently 2 efficient implementations for R and for Python, and hopefully more to come in the future. And it's a very active thread of research in statistics. So I think you can expect more on the matter very shortly. Thank you very much. And if you want the details, it's all been published in this 2018 paper. Thank you very much.
Hello, I'm going to discuss in that talk how to decode with confidence, which means how to obtain statistical control over decoder maps. The setting where I am working is that of cognitive neuroscience, where you probe of cognitive theories by presenting some stimuli in an experimental paradigm and acquiring neuroimaging data like fMRI data to study the impact on brain activity. And at the time of analysis, you want to compare the stimuli or the behavior of the participant to brain activity. And you can do that with an encoding model that will map the stimuli or some characteristics of the stimuli to brain activity, or you can use a decoding model that tries to infer some information about the behavior of the stimuli based on brain activity. What is very important is that both things are very useful. So encoding is maybe more familiar, but decoding is also important. This was a point made by Weishfeld and colleagues in an article in Neuroimage, where they show convincingly how both encoding and decoding are necessary to interpret correctly statistical associations between brain activity and stimuli. So today I'm going to focus on inference when we consider the decoding problem. I will call X the brain data that are recorded, so the brain activation maps, and Y the stimulus or some characteristics of the stimulus that are being studied. In this talk, I will consider the use of linear models to map X to Y. And the reason is that brain activity is represented by many variables, many covariates, many features, and only few samples. I will call P the number of features, N the number of samples. Also, the signal-to-noise ratio is low, and there is no such thing as a translation invariance that would justify using convolutional architectures for that. Finally, it has been shown empirically that linear models still outperform neural networks for decoding tasks. Now, the problem is the following. When you take a given problem, like here in the HAXB dataset, classifying image of face, the response to face images, as opposed to other kind of visual categories, you may have many different models that perform more or less equally well in doing the task, but with completely different internal models. Some of them can be sparse or non-sparse, and this does not depend on the data. This is a prior that is given by you, by the experimenter. And so you have these different weight maps that represent possible maps. And so it's impossible to conclude from that for each brain region whether it's actually involved in the process or not. So we're going to address this question today. And the first thing we need to do is to change a bit the problem. So for that, we will interpret, so if we model the brain as the X variable as a set of signals in different location, X1 to XP, that are used to predict some behavioral information Y. The inference that we are trying to make is to know whether region Xi is adding some information in the prediction of Y on top of the other, which means conditional on the other covariates or features or brain region Xj for J other than I. Is there an association between Xi and Y? Now, if we take a decoding setting, so we see behavior as a function of brain activity, so Y equal XY plus E, XW, sorry, plus E. This amounts to testing whether the component I of the weight vector W is non-zero. So is WI non-zero? So what we're going to do today is to provide some inference method that is to be able to say that WI is non-zero with some statistical guarantees. It turned out that the machine learning community has been developing solutions for this very hard problem in the recent years, like knockoffs or the sparsified lasso, and I will mostly describe dispacified lasso, the use of dispacified lasso today. To understand it, we first need to go to the old classical linear models that have been known since Gauss. So the ordinary least square solution, which is valid as long as the number of observations n is larger than the number of covariates p. And in that setting, you can obtain some weight estimate by the ordinary least square equations together with an estimate of the covariance, so the joint error on this estimate. And then you can test whether feature I is significantly different from 0 using a t-test, typically. And this works as long as x transpose x is invertible, which means that x is well conditioned. Still, we have technically a small problem, which is a multiple testing problem when we want to test all regions I, because the tests are not independent, but let's forget about that for the moment. The problem is that when P becomes as large as n, or even larger, then the solution is no longer defined. Here, I just took an example for illustration where n equal 100, P equal 95. The true model that generated the data is the red line, and the model that we obtain from the data x and y, so our estimate of W by the ordinary least square is the blue line, which kind of has a crazy variance, crazy oscillations. And actually, the confidence intervals about the solution is very hard. So we have very low confidence in what we get. So this is essentially useless. It illustrates the fact that ordinary least squares break when P becomes as large as n. By contrast, the recently proposed dispacified lasso solution is very close to the ground truth and with a narrow confidence interval. This is why we should consider it in high-dimensional inference. It even works when P is larger than n, but not when P is much larger than n. Here, P equal 2000, n equal 100, and we see that the dispacified lasso solution again becomes crazy with this very large oscillation and very large confidence intervals. And so we need to do something else to make it work. And what I propose to do is to reduce the dimension of the data. Let's take neighboring covariates, let's group them together and consider them as one thing, and now solve the problem in a reduced setting. For instance, we could reduce the number of covariates to 100 or 200 and then run the dispacified lasso. This is what we do here. And again, we obtain a reasonably good solution and reasonable confidence intervals around the estimate. So what we are going to do with brain imaging is to first use a grouping of the voxels. So actually we'll use clustering in our case of the voxels into regions to reduce the dimension of the data, and then run the dispacified lasso on this clustered data. So this is a clustered dispacified lasso. The problem that remains is that the solution of that will depend on the clustering. And we know that there is no such thing as a good clustering. There are different possible solutions when you want to consider the data. And so what we propose to do is to not to consider one clustering, but different clustering, and ensemble the corresponding solutions, obtaining the ensemble of clustered dispacified lasso and Claudel. An illustration why we need ensembling. For a given problem, again, the XB data set, if you obtain solutions from different clusterings, you see the impressive amount of variability of the solution depending on the clustering choice, which is bad, of course. So you rather want to aggregate all these estimates into one that represents the consensus across clusterings. And so it's an ensembled or aggregated estimator. So overall, what we're going to do is to create several clusterings of the data run the disparsified lasso estimator on all of them, and finally ensemble the solutions to get a p-value in each location doing the marginal inference. The kind of control that we will have with this p-value is not a strong control in each brain location. It's rather a spatially relaxed control, meaning that we can't say anything around the true support, the regions where there is activity. What we can say is that far from the true support, we will be able to control the occurrence of false positives. So there is a special slack delta. And so in a neighborhood, delta neighborhood of the true support, we do not control anything, but at a distance greater than delta from the true support, we will control the family-wise error rate. So this is a spatially relaxation over the standard family-wise error rate control. So the delta will be defined, will be related to the clustering that is used. But what we show is that on simulated data or hybrid data where we use real data and simulate effects on top of it, we indeed control the delta family-wise error rate with the N-Claudel technique. So it's the red curve here. It's actually conservative inference. But the second good news is that in terms of precision recall, which is a compromise between recovering the true support, which is a recall, and keeping the rate of false detection small enough, which is a precision, the precision recall curve is higher for the N-Claudel techniques than for alternatives, which means that for a given precision or false discovery rate control, we have more true detections. Now, in a more qualitative manner, we can assess the phase validity of this estimator on the HCP900 dataset. So on the motor task, where the motor left versus right foot task, we see that we obtain the expected regions. In the gambling task, where we want to discriminate gain versus loss, also we obtain a nice contrast between amygdala and insula, for instance. Or on the language task, we see in the story versus math contrast, we obtain the language regions versus the math regions. It is also interesting to compare this to what we would obtain with marginal inference, which is the classical inference, statistical parametric maps that you would obtain for the same contrast. And of course, marginal inference is much easier. You have more power to detect the effect because you're not conditioning on the all brain regions. But for the motor task, it makes actually relatively little difference in terms of significant regions. For the gambling task, I would argue that the conditional solution is actually more interesting if you look at the amygdala insula contrast that has been found, that you would not really get with a univariate analysis. In the language task, you see that the conditional inference is much more conservative, which means that univariate inference will give you very wide networks with different level of activation. Even the default mode network seems to be involved, while actually the marginal inference would not consider it as a conditionally important region for that task. Now it's time to conclude. So I have shown you that we could obtain confidence intervals or p-values on decoding maps, which is a great novelty. The inference that we obtain is more specific than classical inference, which means that, of course, it's a stricter criterion to be able to say that a region predicts behavior on top of other brain regions. So expect, in general, more conservative inference. The other price to pay is that we cannot say that each detected voxels is indeed predictive. We can only say that with a spatial accuracy delta, which is typically something like 8 to 12 millimeters. So we have a limited precision in the inference, but it can be good enough for most tasks. We are currently extending this technique to other estimators, so further than linear models, so using logistic regression and nonlinear classifiers. The code is publicly available, and we have also extended it to MEG data for the inverse problem. Finally, I would like to thank my colleagues, especially Jérôme-Alexis Chevalier, Bill Nguyen, and Joseph Salmon. Thank you for your attention.